Concordia University's For Space, thank you for joining us for the next part of today's event. Space Day is the event, but today, at the moment, it's a conversation with AstroYeast. Now, to help situate you, we're streaming to YouTube, we're live from For Space, and we are located on unceded Indigenous lands in Chichage, Montreal. Well, we're running this as a live stream Zoom meeting also, so anyone joining us there, if you have questions, pop them in the chat. Those of you in the space, just raise your hand. We'll bring a microphone over to you, and that will be really great, Q&A. Okay, with that, it's my pleasure to pass things over to the project manager for AstroYeast, Lancia Lefebvre. Welcome back, Lancia.
Hi. Hi. So I'm here on behalf of AstroYeast today. We are a student-led team. Just a little bit of brief history. We started out, can't get my slides to turn now. There we go. So we started out as an undergraduate team as part of the iGEM. It's an international genetically engineered machines competition. This was an undergraduate team where we got to tackle like a real world problem with synthetic biology. And we started exploring a space topic. So could we use yeast for bioproduction in space? And then as part of iGEM, if you want to learn more, go to igem.org, really cool organization. We have to also fundraise to fuel our research. So we applied to the Deep Space Food Challenge as part of that. And we landed the first award as semi-finalists. And then we continued on, and we're one of 4 Canadian finalists in the Deep Space Food Challenge. That just wrapped up this year. We did not win, but congratulations to EcoAtion for that. It was an incredible experience. And then in 2023, so mainly what I'm going to talk about today, Space Concordia came to us because they were applying for the CUBICS grant, so a CubeSat initiative for Canada in STEM. And they were looking for a payload for one of their CubeSats, and so that's what I'm going to be focusing on today. So what do we do as part of the Deep Space Food Challenge? We were interested in bioproduction in space. So making materials or food or medicine in organisms. We focused on yeast because it's a relatively easy organism to bioengineer. It's renewable, so you can send a small amount up and just grow a bunch more. And it's also like generally regarded as safe. So there's yeast in what you're baking bread, and that's kind of similar to the strain that we are working with. So we worked on this yeast strain. We wanted to engineer it so that it could also be an automated system. Astronauts do not have a lot of time when they're in space. So we built a custom bioreactor. Unfortunately, I didn't put a photo of it here. I should have. It looked great. And then we also wanted the bioproduction itself to be automated. So typically, to induce production of nutrients or whatever you want to produce, you'll add chemicals to your mixture to induce the gene expression. So another option is to use light. So that was something we're working with, and I'll talk a little bit more later. So our end food product, in principle, was a yeast paste. So you could eat this yeast paste, you could mix it into with other ingredients, make like a miso soup. We made a crepe out of it, and then it would have flavor or nutrients. So flavor molecules or produce vitamin A, vitamin D, vitamin C, stuff like that. And the benefit of the genetic system is that it's modular. So we could plug and play, like punch in different genetic constructs to produce different vitamins. So for the CUBICS initiative, maybe you guys are probably familiar with what a CubeSat is by now, but so you have this relatively small satellite. We were approached to work with a payload for 1/3 of that satellite. We've been collaborating with the Space Concordia team and a capstone team. It's been super fun, honestly, a really amazing experience for the SC Friar satellite, which is going to be launching in a year or two. So Space Concordia has successfully already sent a CubeSat to space, so we're really excited to be working on the second one with them. So as part of that mission, we want to know, will our yeast strains be viable in space? This has previously been published, so we're kind of confident it's going to stay alive. But mainly we want to know, can we use this system in space to produce nutrients, and can we do it with light? So this is an example of the genetic construct I was talking about. So we have our, it's a optogenetic system essentially composed of these proteins that are in purple. So you have two transcription factors. Just think of them as proteins that are like two half proteins. It comes from a plant. When you shine a light on it, specifically blue light for this system, the two proteins come together, they dimerize, and then they're able to bind the promoter. And then binding the promoter begins the gene expression. And so where you see this like nutrient or flavor kind of box, that's where we can plug or play and like put in different constructs for either nutrients or flavors. So this is what we want to see, that in space, will this work? Can we make it happen? So the two goals, again, viability of yeast in space, and can we induce the production of vitamin A with light? We chose vitamin A. It's a small construct, and it also turns yeast cells orange. So it's really easy to monitor. It's a visual cue. I'll show you more in a minute here, but we'll be using absorbents. So it's really easy to detect because it turns orange. And we figured we could do this in a fluidic card. So this is just a cross-section. But basically, the yeast cells get trapped in a well, and then fluid is able to pass, and the yeast cells are constrained within. So here's basically the fluidic card we're going to be working with. It's just a bunch of layers, polycarbonate mostly, and then there's a couple filter layers that hold the yeast on the inside. Fluid flows in, fluid flows out, and then the most important part is that from top to bottom, it's clear. So we can shine light across it and then detect on the other side. To be transparent, we're very fortunate also. This design is based on Biosentinel. So there's a couple experiments that came before us. Pharmasat was one, and then Biosentinel was built on that. And they sent an experiment with Artemis mission that just launched recently. So they were going to test the effects of deep space, radiation and microgravity on yeast cells, because yeast cells are an analog for human. Unfortunately, their experiment failed. It was left on the tarmac too long. It was too hot, too humid, and they believe that their yeast cells died before they even made it to space. But so this is heavily based on their design. And now compared, to add to that, there are challenges with sending experiments to space. There's extreme conditions. So whether you're in the sun or the dark, there's massive fluctuations in temperature. We have to keep our cells alive. Again, Biosentinel, unfortunately, that didn't happen. And then our observation methods are really limited. So we have a very small area in which we can observe. Like in the lab, we have a lot more equipment we can work with. But in this tiny little box, we are limited by which options we can use to collect data. And so environmental control, and a lot of this taken care of with our collaboration with Space Concordia. So there you see Jonathan Rubin and Jacob, along again with the capstone team. They built this payload capsule and built it in the Concordia makerspace, which if any of you don't know and you're part of Concordia, definitely check it out. It's very cool. So this capsule is pressure tight and airtight. So it'll hold the air in it from the time that we load our experiment until we initiate the experiment. And then you have the payload environment. So the fluidics card sits in the middle. On top, there's an LED layer. So the LED is providing the light that we're going to shine across our sample. The detector is on the other layer. There's a heating plate in there as well. And then also fluid handling. We have to send culture media to our cells so that they can grow. And then data collection. So the data goes out on those wires, goes to the central part of the satellite, CubeSat, and gets sent back to Earth. So just a really general overview. There you have Michael Pagden from Biosentinel. Again, big shout out and thank you to them for all their support. So to overcome this problem of yeast making it from Earth to space is called a desiccation protocol that we're using. It's basically just air dried. So we grow the cells up, we air dry them with a sugar that kind of helps them crystallize and keeps them alive. It puts them in a dormant state. We're then going to assemble this fluidic card with the satellite, so it'll be put in this airtight capsule. The satellite is transported to the launch site. This can be in Florida, so you can begin to imagine like how long these cells are going to be in this dormant state for. This is likely Nanoracks. It can be another commercial provider. It'll be launched in unused rocket space, so CubeSats kind of fill empty space on rockets. And then it'll be received at the ISS currently. We're still slotted for that orbit. And then from the ISS, it gets released into orbit around Earth. And then it'll be in orbit until it crashes into Earth's atmosphere. So as far as the experiment goes, the first thing that is going to happen is that the satellite is going to get launched, the satellite is going to stabilize, and then the satellite is going to get turned on. And once the satellite is powered up and stabilized, we can start our experiment. So the capsule will warm. Yeast likes to grow at 30 degrees Celsius, so it's going to warm up. We're going to make sure the fluids that we have in there aren't frozen. The dormant yeast cells are then going to get rehydrated. They're going to start to wake up, begin cellular metabolism. We're going to give a minute to do that. And then we're going to start collecting data. So the LEDs are going to be shined across. The absorbance principle, we shine light across a sample. It absorbs a certain wavelength, and whatever we can read on the other side based on previous experiments, we can deduce that back to things like cellular growth. We're going to be using an indicator dye for viability that turns from blue to red, so it is reduced with cellular metabolism at specific wavelengths. This again is it's a commercially available dye, but BioSentinel modified it for their experiments, so we're basing off of these papers. And then we're going to shine our blue light, which will initiate gene expression of vitamin A once we've attained enough cellular mass. And using another light, we're going to monitor and see whether we're producing vitamin A and to what extent. So here's an example of the setup. We have our two fluids. The Alamar blue is a viability indicator dye, so it'll tell us to what extent the cells are alive. It has to be kept separate from the media because it'll disintegrate. It essentially degrades when they're combined together. So the two fluids will be mixed. They'll be pumped into the card. There's a waste bag just so that we can flush more media if we need to for long-term experiments. And then the desiccant trap. So very important to keeping our yeast cells dry until we want them to have moisture. The minute moisture hits them, they can wake up. And if they wake up too early and they don't have nutrients, they'll die. And here's just a few more details on the data collection method. So on the right, you see the two graphs for the blue and the pink. So the oxidized and the reduced form of the viability dye. And then at 470 nanometers, so in the bottom right, that's where we're going to detect our vitamin A. There's a typo in the wavelengths, but if anybody wants to discuss that later, I can clarify. I don't think I need to now. So we have the 4 LEDs, so the 850 nanometers, 570, 630, and then our 470 nanometer LED is going to have a dual function of inducing the optogenetic system and also detecting our vitamin A. So in the upcoming months, we have our fluidics card. We've been prototyping. We've actually moved on to kind of the more final materials at this point. You can see the filter more in this one, kind of the circle that's on top that'll keep the yeast in there. Bottom right, we have our LED testing rig that Space Concordia built for us, so we're able to test the system. And then, of course, we're building the strain. But in the next month, we're hoping to bring this all together. So the environment and our fluidics card and our yeast cells to get it going. And then after that, it's just going to be a whole lot of testing until we have the opportunity to launch. So we are supported, of course, by the Canadian Space Agency, Deep Space Food Challenge. A big thanks to SolidWorks Alumni Association as well. Big shout out to Concordia, our supervisors, Orly Weinberg, a big supporter of ours. Space Concordia, of course, a total pleasure to work with them. And yeah, if anyone has any questions or anything you'd like to know, I'm happy to answer questions now. Yeah.
We'll just run a microphone over to you. Hold on one second.
Hello.
Hi.
You mentioned you work with is it the DNA that you modify, or no, the DNA stays the same?
Yeah, so we're editing, right now we're working with a plasmid. If they were to be sent to space, we'd want to integrate it into the genome of the yeast. But yeah, it's modifying the DNA, so taking genes from other organisms and putting them into yeast to produce them. So vitamin A, the genes we're working with actually come from another strain of yeast that produces, I think it's beta-carotene lycopene. So we take these known genes and then insert them into the yeast genome. And then the optogenetic system is coming from plants. So we amplify and take these genes and put them in the yeast genome. And so now our yeast is producing both of these, the optogenetic system, which will turn on the expression of the vitamin A.
Do you have any concerns of like radiation affecting that, or no?
So radiation, Dr. Corey Nislow is one of our consultants, and he's a yeast researcher from UBC. He sent on 2 Artemis missions, I think now, but he did a yeast deletion collection. Basically, he sent a whole bunch of strains of yeast up to space, and they looked at radiation. We're in low Earth orbit, so there's a lot of protection from Earth's magnetic field and whatnot for radiation. And he said that their results, so they delete every gene in the yeast genome just to see kind of what happens, what's essential, but they saw no significant impact on yeast, like gene expression, or no significant impact from radiation. So as far as like operating in low Earth orbit, it's not a concern right now. Obviously, if we go into, I think, like further out away from the Earth, radiation, I think we know is a concern, especially coming back from some of the spaceflight data we're getting right now from astronauts being up there. But there are ways to mitigate this. Like when we were talking about building the bioreactor, you can encase it, you could protect it. Yeast aren't as big as humans, so it'd be easier to protect them from radiation. Yeah. Cool.
So I wanted to ask, how long is the in-orbit process approximately?
I don't actually know that answer. It's like I haven't gotten there yet. I think Space Concordia would know better than me. I think, I don't even know. I'm going to say probably about a month or so. I know it's longer than 2 weeks because we've talked about running the experiment that long. But yeah, that's a good question. Yeah.
Any other questions in the space or online?
You mentioned you made like pancakes and like other stuff. Like, do you have to bring up like the farine up to or do you just need the yeast and whatever
so
that system
we
is?
built that with, we worked with a chef, Laura Feria. So we had a chef come in and build some recipes for us. And so that recipe was made from like mostly yeast and then a little bit of flour, like a small amount of flour. So that's kind of like as our project evolved, we definitely saw our yeast taking on being more of a supplement to other foods as far as like long deep space missions go. Because like you said, otherwise you have to bring up all these other ingredients. One of the successful recipes, I think, as far as like just using yeast though, I was really impressed with the miso recipe because you could just add water and it did give like quite a umami flavor to it. But yeah, with the pancakes, there was a little bit of flour in there. Yeah.
What do you think is the most challenging part of the entire AstroYeast project?
Whew. I guess, honestly, I would say there's a lot of components. So one of the, like, you can think that, you know, like doing experiments in space comes with some unique challenges. And so, like, we start with the genetic engineering, you know, and so that has its own challenges. And so we tackle that. And then we're like, okay, we'll build this fluidic card and we'll base it off of the BioSentinel experiment. But then we need to adapt it for our own needs or fit it into this small capsule, find a way to like adhere to power constraints, and then testing all of these tiny, tiny things. So like testing the genetic system, testing the fluidic system, testing the resilience of the material, the sterility of the material. So everything's gonna have to be super sterile. I think one of the biggest things is going to be keeping the cells dry and alive and sterile. Like, if we can get the cells up there and they're not contaminated and they're in their dormant state, that would be awesome. I think we can get data. So I guess maybe after all that, that's probably the biggest challenge. Yeah.
Do we have any other questions? One more.
Amazing.
Round three. Do you have any like weight constraints, given that you have to launch it up, or how big of a
Yeah.
factor does it play with like today's materials?
Yeah, no, there's absolutely weight constraints. Space Concordia has been taking care of that, like, because everything we're building is quite small and light. But when you apply for these grants, like both grants, so the DSFC award and then the Cubics, they give you constraints and parameters. They're probably publicly available as well. There's also like constraints on what materials you can send up. They have to be space grade, space safe materials as well. Yeah, I don't have those numbers for you. Yeah, but they absolutely exist and are a limiting factor. Yeah. Cool.
One last question? No? We're good?
Cool.
Lancia, thank you.
Yeah, no problem. And if anybody's like interested in space projects, like go for it. It's super fun. It's really worth it. Right now is an amazing time to get into it. There's a lot of opportunities, and like just go for it. Just apply for it. Give it a go. It's worth it.
Just go for it. Yeah. Okay, let's wrap it up. That's great. Thank you again, Lancia.
Thank you.
Okay, everyone, thank you for joining us today. Thank you for joining us for all of the events as part of Space Concordia's Space Day. Those of you joining us online, we're going to be closing up the live stream now. Just remember, this remains available on our YouTube channel if you'd like to revisit or share it there. Thanks, everyone. Thank you, Lancia.